Hello students, I am Shahin Mehmood, currently studying MBBS from College of Medicine and General Hospital, Kalyani, and today I will explain the botany section of FL6 full test 6. So let's start. So first question is given, first question, that in 5 kingdom classification system, which kingdom are unicellular eukaryote? We know that there are several kingdom classifications like 2 kingdom, 5 kingdom, 6 kingdom, and among them 5 kingdom classification was the most popular and it was given by R.H. Whitaker. And there are several groups, that prokaryotic and eukaryotic, and the single cellular eukaryotic group there was Protista. So first option will be our correct answer for this question. Moving towards the next question. Next question is given that agar is used to grow microbes in preparation of ice creams and jellies. The reason is given agar is hydrocolloid which is produced by certain marine brown algae. And we need to choose between all these 4 options. So let's understand this question. So agar is used to grow microbes in preparation of ice cream and jellies. So it is absolutely correct statement because we know that agar like Gelidium and Gracilaria, Gelidium and along with that Gracilaria, these are basically the agars that are used in preparation of ice creams and jellies. So it is a correct statement. Now the reason is given agar is a hydrocolloid which is produced by certain marine brown algae. So basically agar is not a hydrocolloid because it is used in preparation of making ice cream and jelly. So a hydrocolloid, what does hydrocolloid means? Hydrocolloid basically means that water holding substances. So water holding substances, hydrocolloid means water holding substance. But agar is not a water holding substance. Some example of water holding substances are like algae. So algae matches with somewhat brown algae, which is not agar. Algae is not a agar, brown algae. So here assertion is correct, but reason is false. So our answer for this question will be answer number 3rd. Assertion is true, but reason is false. That will be our correct answer for this question. Now moving towards the next question. Lycopodium and Adiantum belongs to the which class of pteridophytes? So both are examples or both are the families of pteridophyte and we need to classify them. First, both Pteripsida, then Psilopsida, Pteropsida, Lycopsida, Pteropsida, Lycopsida and Spinopsida. So Lycopodium you can remember like this way, Lycopodium belongs to Lycopsida. So Lycopodium belongs to Lycopsida, Adiantum belongs to Pteropsida. So Lycopsida and Pteropsida. 3rd option will be our correct answer for this question. So this kind of question, to attempt this kind of question, you need to remember the facts or the information given by NCERT. So these are the kind of remembering type of question. There is no understanding based question in this kind of question. So you need to read NCERT to solve this question. Moving to the next question. Given below are 2 statements. In parietal placentation, the ovule develop on the inner wall of ovule. And the second statement is given when stamens are attached to perianth, they are epipetalous as in lily. So we need to choose between these 4 options. The first statement is given in parietal placentation, the ovule develop on the inner wall of the ovary. Let's understand what does parietal placentation actually mean. So if we want to make a structure, so this is the ovary. This sort of placentation we can see that the ovule, these are the ovule. These are ovule. These are ovule. So these are on the inner side or inner wall of the ovary. So this statement is correct. The next statement is saying when stamens are attached to perianth, they are epipetalous as in lily. So basically when stamens are attached to peri and they are not epipetalous, they are epiphyllous. So that's why this statement is wrong. This should be epiphyllous. Epiphyllous. So our correct answer for this question will be second statement. The statement first is correct, or the statement second is incorrect. Now moving towards the next question. Trilocular ovary is found in Brassicaceae, Solanaceae, Fabaceae or Liliaceae. So basically we need to understand that what are the locules in ovary. So these are ovary, and if trilocular ovary is asking, so basically these are the locules in ovary. These are locules in ovary. So trilocular ovary is found in the family of Liliaceae. 3 locules or cavities in it. And what are the others? So when we are solving a question, attempting a question, we need to also understand the incorrect options as well, why it is incorrect. So if you are doing this, then you can sometimes eliminate other options as well. So you need to understand. So in Brassicaceae, that is bilocular. Where is that? Bilocular. In Solanaceae, it is also bilocular. In Fabaceae, it is unilocular. In Liliaceae, as you already said, it is trilocular. Okay. So fourth option will be our correct answer for this question. Moving towards the next question. Jute is so what jute is? Bast fiber from secondary xylem, bast fiber from primary xylem, bast fiber from secondary phloem, bast fiber from primary phloem. So jute fiber is also known as bast fiber obtained from plant or skin of the secondary phloem. So third option will be our correct answer for this question. Now moving towards the next question. Given below are the two statements. One is level assertion and reason. The assertion is given, cell also vary greatly in their shapes, and reason is given, the shape of cell may vary with functions they perform. So if we put because here, because you can see that now we can read the statements. It is saying cell also vary greatly in their shapes because the shape of cell may vary with the functions they perform. It is absolutely correct. Assertion is also correct, reason is also correct, and it is satisfying both, explaining both. Why this is satisfying both? Suppose there is a cell, we know that cells have various size, various kind of size. One is big, one is small, one is. So why this is big and why these are small? So a cell in our ear may be small, and a cell in our leg could be big. So this is an example of human cells. If you example a plant cell, a cell in leaf could be small, and the same cell in other parts of the plant could be big. That is because the functions they perform. So they have to, if they have a bigger functions to perform, then they have a big amount of cell, big amount of cytoplasm, big amount of nucleus. And if they have to perform less complicated function, they have smaller cells. That's why cells vary in different shape and size, because they of the functions they perform. So both assertion and reason is correct, and assertion is the correct explanation. Reason is the correct explanation of the assertion. The fourth option will be our correct answer for this question. Moving towards the next question. Which of the following is the incorrect statement about prokaryotic ribosome? Prokaryotic ribosome you have to find. Ribosomes is the site of protein synthesis. Yeah, it's absolutely correct. You know that ribosome is the machinery of protein synthesis. Second, 50S and 30S subunit to form 70S ribosome. 50 and 70 will form 70S ribosome. 50 and 30. So it is absolutely correct. Now the next, 60 and 40 subunit unite to form 80S ribosome. That is basically not happening. It is a correct statement, but this is not happening in prokaryotic ribosome. This is basically happening in eukaryotic ribosome. That's why this question is asking about prokaryotic statement. This is the incorrect statement. Third option will be the correct answer for this question. Now moving towards the next question. 109. Crossing over. So we need to find the correct option about crossing over. Crossing over is saying non-enzyme mediated process, first option. Second option, not exchange of genetic material, and an enzyme mediated process, third option, none of these. So what is crossing over? So you know that crossing over is basically the exchange of genetic material. It happens like this. If we want to show somewhat. See. So this part where both chromatids are exchanging their arms, this is called crossing over, and it is a enzyme mediated process. The enzyme used here is recombinase. So 190, the crossing over is the enzyme mediated process. Third option will be our correct answer for this question. Now moving towards the next question. The cell divide to restore the mitochondrial cytoplasm ratio, nuclear cytoplasm ratio, ribosome cytoplasm ratio, or ER cytoplasm ratio. So we know that if a cell, this is a cell and this is a nucleus. Always we consider the nucleus and cytoplasm ratio, not mitochondria, not ER endoplasmic reticulum, or not ribosome. It is always associated with nucleus and cytoplasm, which is also called as MC ratio. So second option will be our correct answer for this question. A cell divide to restore nucleus cytoplasm ratio, nucleolar cytoplasm ratio. So the correct sequence of one, and then moving to the next question, it is saying that the correct sequence of cell cycle. So correct sequence of cell cycle, let's draw it. So if we draw, you can see that this is G1 phase of cell cycle, this is S phase of cell cycle, this is G2 phase of cell cycle, this is M phase of cell cycle. So how it will? It will be G1 phase of cell cycle, then S phase of cell cycle, then G2 phase of cell cycle, then M phase of cell cycle. So third option will be our correct answer for this question. Moving to the next question, moving towards the next question. Recombination is completed by which stage in meiosis? So basically these are the stages of prophase 1, leptotin, zygotin, packaging. Prophase 1. So leptotin, we know the first stage, then zygotin, then packaging. Leptotin basically the bucket stage, and zygotin has synaptonymal complex form. Synaptonymal complex is formed. In packaging, recombination is completed. So third option will be our correct answer for this question, leaving the chromosomes linked at the site of crossing over where recombination is completed in packaging stage. Moving towards the next question. What will be the number of Calvin cycle to generate one molecule of hexose? And the options given 8, 9, 4, 6. So Kelvin cycle is also known as C3 cycle, and we know that in one cycle of Kelvin cycle we can induce or we can obtain one molecule of glucose. One cycle, one glucose. So here 6 cycle is needed to obtain 6 amount of glucose or hexose. Is the same thing basically. So any kind of sugar you need to obtain. So fourth option will be our correct answer. 6 cycles of Kelvin cycle is needed to produce 6 moles of hexose. Fourth option will be our correct answer for this question. Now moving towards the next question. 140. The hilum is a scar on the fruit, seed, or fruit. And the options are varying. That first option is given fruit where style was present. No, it is not a correct answer. Then second option is given seed above where a small pore called micropyle was present. It is absolutely correct answer. Second option will be your correct answer. Suppose this is a seed. Then see here is this is the hilum. This is the hilum, and this is basically the micropyle. This is basically the micropyle, and where above it hilum is present. So basically this is the hilum is a scar on the seed above where a small pore called micropyle was present. The third, seed where funicle is attached is not correct, and fruit is also not correct. Second option will be our correct answer for this question. So I will basically present on seed with a kind of score. So moving towards the next question, 150. It is saying, which of the following fruit is parthenocarpic? So what does parthenocarpic fruit means? Parthenocarpic fruit means so there is no fertilization. There is no fertilization. If we read the explanation, that fruits, parthenocarpic fruits are the fruits which are formed without fertilization. These fruits are naturally seedless. So you can see the example of parthenocarpic fruit is banana. Second option will be our correct answer for this question. Moving towards the next question, 160. ABO blood groups in humans are controlled by gene 1. It has 3 alleles, 1A, 1B, and I. Since there are 3 different alleles, 6 different genotypes are possible. Yes. How many phenotypes can occur? So phenotypes can occur A, B, AB, and O. Basically, these are the 4 phenotypes that can occur. So our answer for this question will be 3. That means 4 phenotypes can happen with this combination. So third option will be our correct answer for this question. So when you are solving this kind of question, during my time, I used to revise a lot. I used to read NCERT a lot of times. So when you are attempting a full test, at least try to remember the main important points. Revise the NCERT. You can't revise the whole NCERT in one night or one day, but try to at least remember the basic points. These are the things like you can attempt maximum number of questions, because you can see the trend is that maximum question you will get from NCERT. Out of 100 questions in biology, which you have to attempt 90 questions, you will get 7 to 8 questions from the NCERT itself. I scored 360 out of 360 in my time, so you can score that too. There is no problem. I was always scared that I am sharing one of my stories that during my time what happens that when I was attempting mock test, I was always scoring below 300 in biology. I was never assured of scoring full marks. Like I am not assured even 320, but I scored full marks in biology. That is because I repeated, revised multiple times, and the questions which you are making wrong, make a notebook and try to revise one more time that where I did wrong, and then you will improve. That's how it works. Like if you are not revising, if you are just attempting the test and then and scoring the marks, that what marks you are getting, then it is of no use. The test is of no use. अगर आप लोग ऐसे करते हो कि टेस्ट देके फिर गलतियां ढूंढते हो कि कहाँ गलती किया, उसको सुधारते हो, फिर टेस्ट का मतलब होगा कुछ. अगर 10-15 फुल टेस्ट दे रहे हो, उसका बेनिफिट भी आना चाहिए. ऐसा नहीं कि टेस्ट दिया मार्क्स देख लिया हो गया, ऐसा नहीं करना चाहिए. आप लोग पढ़ते रहो, टेस्ट देते रहो, डाउट पूछते रहो, सवाल देखते रहो, जैसे एक्सप्लेनेशन होता है, कैसे कैसे क्वेश्चन का अप्रोच करना चाहिए. ऐसे करते रहोगे तो बायोलॉजी में 350 आपका कहीं नहीं जा रहा. ओके, सो मूविंग टुवर्ड्स द नेक्स्ट क्वेश्चन. So these are the things you have to remember. Pick out the correct statement. 170. चीजों का अगर ज्यादा पढ़ोगे तो ज्यादा देर तक याद रहेगा, लंबे समय तक याद रहेगा, क्योंकि बायोलॉजी में हर चीज समझना नहीं होता है. कुछ कुछ चैप्टर होता है जहाँ पे आपको याद करना ही करना होता है. वहां पे अगर आप याद नहीं करोगे तो आप नहीं समझ पाओगे. पहले आप याद करो, फिर देख लो कि जैसे प्लांट मॉर्फोलॉजी हो गया, इकोलॉजी हो गया, या फिर इंफॉर्मेशन डेटा आपको याद रखना है. याद रखोगे तो इजिली क्वेश्चन हो जाएगा. जिस चैप्टर में समझना है, जैसे जेनेटिक्स हो गया, फिर डीएनए फिंगरप्रिंटिंग, ये सब जो चैप्टर्स है यह हो गया, तो यहाँ पे अगर आप करोगे, ऐसे करते रहोगे तो इजीली आपका नंबर आएगा. सिर्फ क्वेश्चन अटेम्प्ट करने से नंबर नहीं आएगा. क्वेश्चन जो अटेम्प्ट, अगर आपका नंबर अच्छा आ रहा है तो बहुत बढ़िया बात है, आप सही रास्ते में चल रहे हो. अगर नहीं आ रहा है तो भी कोई बात नहीं, आप कोशिश करते रहो बस. 117. moving towards the next question. So pick out the correct statements. Hemophilia is a sex-linked recessive disease. It is a correct statement. Down syndrome is due to extra 21 chromosome. We know that trisomy 21. Down syndrome. Trisomy 20. That is the correct statement. Now the next statement is given that phenylketonuria is an autosomal recessive gene disorder. Phenylketonuria is correctly an autosomal recessive gene disorder. The next statement, sickle cell anemia is an X-linked recessive gene disorder. Sickle cell anemia is not an X-linked disorder. It is the autosomal recessive disorder. Autosomal recessive. Sorry, there is not autosomal recessive. I'm writing it. Autosomal recessive. तो या सो वहाँ पे हम लोग देखेंगे तो क्या क्या ऑप्शन हमारे which options are correct. First, second, and third options are correct, and fourth option is wrong. So first, second option will be our correct answer for this question. 1, 2, 3 are correct. Moving towards the next question. 180. A pleiotropic gene. What does a pleiotropic gene means? First of all, you have to understand when approaching a questions. Don't just first of all read the options. Try to understand what does this term means. Pleiotropism. So what does pleiotropism means? The ability of a gene to have multiple phenotypic effects because it influences a number of characters. That is the meaning of pleiotropism or pleiotropic gene. Now read the options. Controls a trait only in combination with another gene. This is not correct. Controls multiple traits in an individual. This is the correct option I am saying, but read all the options. Is expressed only in primitive plants. It is not correct. It is a gene evolved during Pliocene. This is the evolution. No, but this is not correct. This is the second option will be our correct answer. And for example, you can take the example of phenylketonuria as well as sickle cell anemia. This control more than two characters or more than one character. That's why it is called pleiotropic gene. Pleiotropism. Now moving towards the next question. Which of the following pairs of nitrogenous bases of nucleic acid is wrongly matched with category mentioned in this? So you need to find the wrongly matched. Guanine, adenine, purine. So one trick I will give here. So during my time, our biomolecules sir also keep the same trick that आग अगर लगानी है तो पूरी लगाओ. So purine, suppose this is purine. So adenine and guanine, this is AG, AG, AG. And then pyrimidine will be another three. Cytosine, uracil, thymine. Cytosine, uracil, thymine. So if you remember this, then you can also remember this because this is basically the opposites. So this is purine and this is pyrimidine. Now let's check. Guanine, adenine, purine. Adenine and thymine. So you can see that. Adenine and thymine. There's only two are purine. Thymine is not a purine. It's pyrimidine. Second option is our wrong. Nitrogenous base match. So these are correct answer for this question. Thymine and uracil, pyrimidine. Uracil and cytosine, pyrimidine as well. So second option will be our correct answer for this question. Moving towards the next question, 120. Semi-conservative replication of DNA was first demonstrated in. The semi-conservative nature first demonstrated in Escherichia coli or E. coli, and it was done by Mendelssohn and Stoll in 1985. 1980, not 85, 1958. Mendelssohn and Stoll. First option will be your correct answer for this question. The other options given are basically used for different experiments. That's given in NCERT. Towards the next question, 121. What could happen if in a gene encoding a polypeptide of 50 amino acid, 25th codon UAU is mutated to UAA? Now read the options. A polypeptide of 24 amino acids will be formed. A polypeptide of 24 and 25 amino acids will be formed. A polypeptide of 49 amino acids will be formed. A polypeptide of 25 amino acids will be formed. We know that to form an amino acid we need 3 nitrogenous bases. Suppose this is AUG starting codon. Then it happens like this. So this is the first amino acid. This is the second amino acid. This is the third amino acid. And at the last one UAA is formed. This is the stop codon. This will not form any amino acids. So before that, 24 sets of amino acid will be formed. So first option will be our correct answer for this question. And you need to remember the stop codon UAA, UAG, and UGA. Must remember this to solve this kind of question. Moving towards the next question. Which of the following RNAs should be most abundant in animal cell? We need to focus at animal cells. So the order of RNA present, that is rRNA, it present approximately 70 to 80%. Then the next is tRNA. Next is tRNA. And last is mRNA. So you can also remember RTM. It's like this: rRNA most 70 to 80%, then tRNA 10 to 15%, and then mRNA 2 to 5%. What is this mRNA? miRNA is basically that microRNA that is 21, 22 base pair long, and these bring degeneration of mRNA. So which of the following RNA most abundant is rRNA? So fourth option will be our correct answer for this question. Moving towards the next question. Decline in the population of Indian native fishes due to introduction of Geriapinas in river Yamuna can be categorized as. Basically, these are examples of alien species invasion. So when there is invasion of another kind of species which are not indigenous, then there is a threat of extinction of our indigenous species. So alien species invasion will be our correct answer for this question. Not co-extinction, not habitat fragmentation, not overexploitation. So that's why I'm saying, अगर आपने इकोलॉजी अच्छे से पढ़ा है, चैप्टर्स अच्छे से पढ़ा है, तो वहाँ पे सारे लाइन लिखा हुआ है, और तो सारे क्वेश्चन एनसीईआरटी से है, लेकिन ये डायरेक्ट क्वेश्चन नहीं है, ये सब क्वेश्चन को आपको मिस नहीं करना है, और ज्यादातर क्वेश्चन सही आ रहे हैं अभी, तो आपको इसमें ज्यादातर ध्यान रखना. Now group of family is included. Next question 124. Group of order is included. Family, order, class, species. अगर आपको इस टाइप का क्वेश्चन सॉल्व करने में कोई भी कंफ्यूजन आए, so just I have a trick. KPC of GS. KPC of GS. K is for kingdom, P is for phylum, C is for class, O is for order, and then F is for family, G is for genus, S is for species. If you say group of order, so group of order will be class. Third option will be our correct answer for this question. Moving towards the next question 125. Given below are two statements. Many plants produce and store chemicals that make the herbivore sick when they eat it, inhibit feeding or digestion, disrupt its reproduction, or it even kill it. And the second statement is saying the plant produces highly poisonous cardiac glycoside, and that is why you never see any cattle and goats browsing on this plant. These are also same correctly pasted from NCERT. So if you read NCERT, you can solve this question. So many plants produce and store chemicals that make the herbivore sick when they eat, inhibit feeding or digestion, disrupt its reproduction, or even kill it. It's correct statement. And the next statement is that the plant produce highly poisonous cardiac glycoside. That's why you will never see that any cattle or goats browsing on this kind of plants. It is also a correct statement. Both statements are correct. So fourth option will be our correct answer for this question. Moving towards the next question 126. We need to match the column A on column B. Parasitism, mutualism, commensalism, and amensalism. You know the meaning of all this. Parasitism means one get harmed and one get benefited. Mutualism means both get benefited. Commensalism basically means that one is getting some benefit, other is getting nothing. Amensalism means one is getting harmed and other is getting nothing. So these are the examples. These are the examples of NCERT. If we match them, 126. Parasitism is basically in Coscuta. Mutualism is basically in pig and wasp. And commensalism is basically in epiphyta and orchid. And amensalism is in black walnut. So the answer for 126 matching all this will be option number 4. Fourth option will be our correct answer for this question. Moving towards the next question 127. Mycorrhiza. Mycorrhiza are the example of amensalism, antibiosis, mutualism, or fungistasis. So mycorrhiza is basically an association between fungi and root of higher plants. Association between fungi and roots of higher plants. And the fungi helps the plant in absorbing various minerals and nutrients from the soil. What plants do, roots of the higher plants do in turn, that it provides the fungi with energy yielding carbohydrates. So both are getting benefited. So what example it could be? Think about it. What example it could be? It is example of mutualism. Third option will be our correct answer for this question. Moving towards the next question. Match the following columns. 128. Plant growth promoter and plant growth inhibitor, auxin, gibberellin, abscisic acid, cytokinin. We know that plant growth inhibitor is gibberellin. Rest all are plant growth promoters. So you can see the options is given 128. Fourth option. First, second, and fourth. That is auxin. Plant growth inhibitor is not gibberellin. It is abscisic acid. So 1, 2, and 4 are the plant growth promoter, auxin, gibberellin, and cytokinin, and plant growth inhibitor is abscisic acid. The fourth option will be our correct answer for this question. Moving towards the next question. Cells derived from root apical and shoot apical meristem and cambium differentiate and mature. So cells derived from root apical and shoot apical meristem and cambium, these are differentiating to perform a specific function. These are called dedifferentiation, differentiation, redifferentiation, or dedefinition. What could be the answer for this question? You need to understand. Mentioned here for the parts root apical, shoot apical, meristem, and cambium, these are basically the primary cells. That's why when they are getting maturation, they are running towards maturation, then it will be called as differentiation. Second option will be our correct answer for this question. Moving towards the next question. A patient brought to a hospital with myocardial infarction is normally immediately given penicillin, is an antibiotic. A streptokinase is a clot buster. Cyclosporin is an immunosuppressant. Then statin is a cholesterol lowering agent. Hypocholesterol lowering agent. So when a patient has myocardial infarction, then what could be done? So to remove the clots, we need to use streptokinase because it is a clot buster. Second option will be our correct answer for this question. Streptokinase is basically a tissue plasminogen activator. It is an enzyme obtained from the culture of some hemolytic bacterium, Streptococcus, and which is modified genetically to remove the clots or clearing blood clots inside the blood vessels through dissolution of intravascular fibrin during myocardial infarction. That's why second option will be our correct answer for this question. Moving towards the next question, 131. In our stomach, the lactic acid bacteria play a very beneficial role in production of butter from milk, prohibit diseases causing microbes, digestion of casein of milk, and lastly neutralization of HCl of gastric juice. Just think about it. The question, lactic acid bacteria. What does lactic acid bacteria do? Production of butter from milk? No. Prohibit disease causing microbes? Yes, it can, because lactic acid bacteria provide check and balance in our gut. That is the correct answer. Second option. Now also we will read the other options. Digestion of casein of milk. Lactic acid bacteria doesn't help in digestion of casein of milk. It is basically in our children have this kind of enzyme which help in digestion of casein of milk. Then neutralization of HCl of gastric juice. Lactic acid bacteria does not have any alkaline property that it will neutralize the hydrochloric acid of gastric juice. So basically these are all wrong. Second option will be our correct answer for this question. Moving towards the next question. 132. So basically, scavengers, detritivore and parasites are given, and we need to match with this column 2: bacteria, fungi, vulture, termites. So what are scavengers? Scavenger basically means that feed on dead or injured animals. So dead or injured animals, examples vulture and crow. And what are detritivores? Detritivores basically feed on dead or decaying matter, feed on detritus. So these are termites and earthworm. And parasites, we know that harm in that produces any kind of harm in our body, and these are basically provide or what can we say, that they give harm by taking the nutrition source of the host. So bacteria and third is matching all this, we will get that second option will be our correct answer for this question. Moving towards the next question. 133. The rate of formation of new organic matter by a cow in grassland is called net productivity, secondary productivity, net primary productivity, gross primary productivity. So let's understand the food chain here. That a grass that is taken by cow. Basically, the grass here is the primary producer and cow here is secondary consumer. So that's why it will be called as secondary productivity. Second option will be our correct answer for this question. Moving towards the next question. 134. Match the following. So P700 is basically PS1, and then it is basically the cyclic phosphorylation. P680 is PS2, and P680 and 700 is basically non-cyclic phosphorylation. Non-cyclic phosphorylation, know that as zigzag pattern. That is basically the non-cyclic phosphorylation when both P680 and P700 acts. If we match all this, we will get will be our correct answer. Third option. P700 helps in PS1 as well as cyclic photophosphorylation. P680 only helps in PS2, activation of PS2, and P680 and P700 both acts when that is non-cyclic phosphorylation. Moving towards the next question. Match the following: vinblastine, ricin, codeine, and gum. So this question is simple to answer. Vinblastine is basically a drug. Ricin is a poisonous thing. Ricin is basically a poisonous thing. Poison. Then codeine is basically an alkaloid, and gum is basically a polymer. If we match all this, we will get our answer 135. Our answer will be our correct answer. These are basically a table from biomolecules. You need to remember each and every table of biomolecules. It's very important. Now moving towards section B. The process by which anything is grouped into convenient categories based on early observable characters is nomenclature, identification, characterization, or classification. So while solving this question, that it is saying the process by which anything is grouped. Basically, three things you have to remember. There is also, I have a trick. I, N, C. First of all, you have to do identification. Then when you have done identification, then you have to name the plant or animal, whatever it is. So you have to nomenclature. Then you have to classify them into any kind of groups or whatever it satisfies. So first identification, then nomenclature, then classification. So this is saying, the question is saying the process by which anything is grouped. So when you are grouping, that means you are classifying into convenient categories. So here fourth option will be our correct answer for this question. Moving towards the next question. 37. So given below are the two statements. Statement 1, isogamous type of reproduction with mobile gamete is present in Spirogyra. Second statement, genera fern also known as homosporous. So isogamous basically means that the gamete will have similar shape and size. Male and female gamete will have both are morphologically similar. And then it is saying that isogamous type of reproduction with mobile gamete present in Spirogyra. In Spirogyra, not mobile gametes, basically non-motile gametes are present because they don't have flagella. Non-motile gametes and non-mobile gametes are present. So this statement is absolutely wrong. Second statement is saying that genera fern is also known as homosporous. Homosporous means the spores have one fixed shape and size. And sporus is basically a form of gametophyte, as we can say that it is basically the production of gametophytes. So this statement is correct. So first statement is incorrect, and second statement is wrong. Sorry, second statement is correct. Sorry, statement 1 is incorrect, but statement second is correct. Third option will be our correct answer for this question. Moving towards the next question. A lateral branch with short internodes, each node bearing a rosette of leaves and tuft of root in water is mint and jasmine, Christian Icornia, banana, or Zamington. So let's understand this. So suppose if you want to make a structure from the information given, you will just make a structure like this. Sorry, it's bad. Basically, these are the leaves and these are short internodes. And below here it is basically a stem, and this part where it will connect another this kind of structure, this whole structure, this will be called as offset. So this whole structure you can see that is tuft of roots. So this kind of things, the lateral branch with short internodes, you can see the short internodes, and each node bearing a rosette of leaves. This is the not a normal leaf, it's a rosette of leaves, and tuft of root is present in Pistia and Icornia. Second option will be our correct. Moving towards the next question. So 130. So in 139, vascular bundles is conjoined, open, and enlarged protoxylem is found in. So we have to make this is present in monocot root, dicot stem, or monocot stem. So here this is phloem, where it will have phloem. It is xylem, and this the structure present between phloem and xylem is basically cambium. So that's why we are calling it open, because cambium is present between phloem and xylem. So, and along with that enlarged protoxylem, this is found in dicot stem. Third option will be our correct answer for this question. Moving towards the next question, 140. Large number of vascular bundle found in monocot stem is surrounded by. So what is the vascular bundle look like in monocot stem if this cambium is not present? Suppose this cambium is not present, it will be closed. So that will be conjoined closed vascular bundle. So conjoined closed vascular bundle found in monocot stem surrounded by collenchymatous multilayered sheath, no. Sclerenchymatous bundle sheath, correct. Parenchymatous single sheath, not correct. None of this not correct. Second option will be our correct. Sclerenchymatous bundle sheath present in monocot stem. Second option will be our correct answer for this question. Moving towards the next question, 141. That is saying that what is the percentage of lipid and protein present in human erythrocyte, that is RBC, red blood cell, present respectively. That is the lipid percentage is lipid percentage is 40% and protein percentage is 50. So third option will be our correct. It's totally informative question. So you can take this. Lipid is present 40% and protein is cellular composition basically. Third option will be our correct answer for this question. Moving towards the next question. 142. So if you remove the fimbriae from the bacterial cell, which of the following would you expect? So fimbriae is basically a supporting element. It is not used for locomotion because flagella is used for locomotion. And then it will happen to the bacteria could no longer swim. It is not correct because it is nothing related to transportation. And then the bacteria would not adhere to host tissue. I am saying this is the supporting element. It supports the framework structure. That's why the bacteria would not adhere to host tissue will be our correct answer for this question. And the third option is saying transportation of molecule across the membrane would stop. Fimbriae is not associated with transportation of any kind of molecule. The shape of bacteria would change, shape of bacteria would not change. That is cytoskeleton structure is different. That is no role with fimbriae. So second option will be our correct answer for this question. During mitosis, endoplasmic reticulum and nucleolus being disappear at early prophase, late anaphase, early metaphase, or late anaphase. You can see that endoplasmic reticulum and nucleolus began to disappear in early prophase. Like you can see the early prophase at some kind of disintegration of all organelles start happening. So first option will be our correct answer, that during mitosis, endoplasmic reticulum and nucleolus began to disappear. Early prophase. Moving towards the next question. Statement-based question. You can see there are so much statement-based question. That means it will consume time. If you have to read, you need to read one time. Like if you are reading one time and then continuously answering it, at least you need 30 or 40 seconds to answer this. So if you read NCERT multiple times and you have practiced maximum amount of question, just when you are giving the actual NEET paper, this will appear similar. This will reflect, and you can answer it like in electric speed. So you know that the immense importance of giving test series. In MEMONEET, experts prepare a very good amount of test series that you can expect maximum number of questions from the test series itself, the full test series that is you are giving now. So okay, the question, answering to the question answer. So consider the following statements and select the incorrect set of option. Cell wall of algae made of cellulose, galactans, mannans, and minerals like calcium carbonate. We know that cell wall is present in algae, then plants. Animals don't have cell wall, and algae have different sort of components, and plant have different sort of components. The components given are cellulose, galactan, mannan, and minerals like calcium carbonate are the composition of algal cell wall. So that is the correct statement. Second, cell wall and middle lamella may be transversed by plasmodesmata. It is the correct statement that plasmodesmata and cell wall can be transversed. Middle lamella and cell wall can be transversed by plasmodesmata. Correct statement. Secondary wall formed on the inner side of the cell. It is also correct statement. So suppose a cell wall, this is suppose a primary cell wall. When the secondary cell wall will form, it will form the inner side of the cell, the secondary cell wall. This one is the secondary and this one is the primary cell wall. Cell wall, another next D statement. Cell wall is a living rigid structure. Cell wall is not living. Cell wall is a dead rigid structure. Cell wall is a dead rigid structure. So incorrect set of option, not only D. Fourth option will be your correct answer for this question. Moving towards the next question, 145. So in cereal grain, the single cotyledon of embryo is represented by coleoptile, coleryza, scutellum, and profile. So basically, the cotyledon are known as seed leaves, and they are attached to the embryonic axis. Dicots typically have two cotyledon, and monocot have one cotyledon. A single shield shape is kind of this kind of shape, shield shape cotyledon that is present cotyledon in grains is known as scutellum. So third option will be your correct answer for this question. Moving towards the question 146. Independent assortment of gene does not take place when genes are located on the homologous chromosome, genes are linked and located on the same chromosome, genes are located on the non-homologous chromosome, or all of these. Suppose independent assortment, suppose this is the pain. So independent assortment means it will not depend on any kind of factor. It will assort independently. It doesn't depend on this chromosome. It doesn't depend on this chromosome. So when this will not happen, when this will not happen, it will not happen when there is a linkage on the same chromosome. That the genes or the chromosomes are linked. That's why it can't assort independently. The second option will be your correct answer for this question. Second option. You can also understand by the explanation saying that principle of law of independent assortment is applicable to only those factors or genes which are present on different chromosomes. It is not applicable on the genes present on the same chromosome, because most of the thing you can see that the chromosomes or genes present, the genes present on the same chromosome that are linked, because crossing over is indirectly proportional to the distance. Now moving towards the next question, test cross. A test cross is carried out to determine the genotype of a plant at F2, predict whether two traits are linked, assess the number of alleles of a gene, determine whether two species varieties will breed successfully. The first option will be the correct answer. We know test cross, basically the name suggests itself, test cross. We just want to find, हम किसी एलिल को पता करना चाह रहे हैं. सपोज एक प्लांट है जो दिख रहा है हमें, सपोज इस प्लांट है, एक प्लांट, ये भी टॉल है, ये भी टॉल है, लेकिन हम इसको नहीं पता कि F2 में ये हेटेरोजाइगस टॉल है, होमोजाइगस टॉल है, F2 में इसका प्रोजेनी क्या आएगा, इसलिए हम इसको क्रॉस करवाते हैं टेस्ट क्रॉस से, ताकि हमें पता चलता है F2 में अगर ये हेटेरोजाइगस होगा तो एक प्रोजेनी क्या होगा, शॉर्ट होगा, एक टॉल होगा, एक ड्वार्फ और एक टॉल होगा, और अगर होमोजाइगस होगा तो दोनों ही प्रोजेनी टॉल होगा. सो बेसिकली हम चेक करते हैं. तो फर्स्ट ऑप्शन विल बी योर करेक्ट आंसर फॉर दिस क्वेश्चन. मूविंग टुवर्ड्स द नेक्स्ट क्वेश्चन, मैच द फॉलोइंग. 148. So RNA polymerase 1, RNA polymerase 2, and RNA polymerase 3. So RNA polymerase, we know the function of RNA polymerase is synthesis RNA by a following strands of DNA. That's the function of RNA polymerase, RNA polymers. And now we have to match with RNA polymerase 1. So RNA polymerase 1 is rRNA. RNA polymerase 2 matches with hnRNA. And RNA polymerase 3 matches with tRNA. So the correct option for the following will be third option, that means C option will be your correct answer. Moving towards the next question. In eukaryotic cell transcription, RNA splicing and RNA capping takes place inside the ribosome, nucleus, dictosome, ER. So remember that when you are reading genetics, most of the things will happen, most of the things happen inside nucleolus, and rest are in cytoplasm. There will be no role of ribosome, dictosome, or ER. Yeah, there are several kind of things that happens in endoplasmic reticulum and ribosome, but most of the things that will happen or that will occur inside the nucleus. So second option will be your correct answer for this question. Now let's understand the question accordingly, that in eukaryotic cell transcription. So unlike in prokaryotes where transcription and translation takes place in the same compartment, in eukaryotes there is a compartmentalization is there. We have read this in cell division or in cell chapter. In eukaryotes, primary transcript is first processed in the nucleus and then transported outside the nucleus. Since the primary, suppose this is the nucleus, this is will be transported cytoplasm. So contains, since the primary transcripts of the eukaryotes contain both expressing gene exon and non-expressing gene introns. Introns will be removed here. It undergoes splicing of introns and later capping and tailing at 5' and 3' occur respectively. So second option will be your correct answer according to the question. Moving towards the next question, that which of the following both pairs have correct combination, in situ conservation or ex situ conservation? What does in situ conservation means? In situ conservation means जहाँ पे कोई एक ऑर्गेनिज्म को प्रोटेक्ट किया जा रहा है जो उसका नेचुरल हैबिटेट है. एक्स सिटू मतलब क्या होगा, उसको नेचुरल हैबिटेट को उठा के लेके आके, मतलब उठा के आने के बाद एक प्रोटेक्टेड जगह पे उसको प्रोटेक्ट किया जा रहा है, दैट इज एक्स सिटू कंजर्वेशन. सो सीड बैंक, in situ conservation, seed bank. No. In situ conservation is not seed bank. Seed bank is basically ex situ conservation. National park is in situ conservation. Option saying in situ tissue culture. Tissue culture is not in situ conservation. It is basically ex situ conservation. Ex situ conservation, sacred groups. Sacred groups is in situ conservation. In situ conservation, national park, correct. Ex situ conservation, botanical garden. It is also correct. Third option will be your correct answer for this question. Let's read the last option as well. In situ conservation, cryopreservation. Cryopreservation is ex situ conservation, and ex situ conservation is wildlife sanctuary. So third option will be your correct answer. So this concludes the explanation of full test 6 of botany part. Thank you for attempting the test. So remember one thing. So lastly, I will say some things before ending the session, that you have to, when you are giving test, don't just, I have also discussed it previously, that don't just give test. You have to analyze it. You have to see why I am wrong, why it is wrong at that time, why it is incorrect. So that's why I will improve. You just don't attempt the test and check the marks and just like go with the flow. It will happen, it will not happen. So suppose in this test, I am saying, suppose in this test you are scoring above 340 plus, then you are going good. If you are below 340, you are not, you have to improve. You just don't get demotivated, but you have to improve. यहाँ पे जो क्वेश्चन से सारे एनसीईआरटी से उठा के लिया हुआ है, यहाँ पे आप एक्सप्लेनेशन पे देख सकते हैं, तो अनशन सवाल एक भी नहीं है, सारे लॉजिकल और एनसीईआरटी बेस्ड सवाल है जो कि नीट में आ रहे हैं. अगर यहाँ पे आपको 340 आ रहा है इस क्वेश्चन में तो आप सही जा रहे हैं, और अगर आपको नहीं आ रहा है तो डिमोटिवेट होने का कोई बात नहीं. आप टेस्ट देते रहिए, मेमोनेट की टेस्ट देते रहिए और आप स्कोर को इम्प्रूव करते रहिए. अब जस्ट ऐसे नहीं कि अगर 220 आ रहा है या 230 आ रहा है बायोलॉजी में, जियोलॉजी बोटनी मिला के, तो आप डिमोटिवेट हो रहा है. डिमोटिवेट नहीं, आप और पढ़िए, एनसीईआरटी डाउट पूछिए, देखिए वीडियोस, लेक्चर्स देखिए, सारे मिलकर आपको एक आइडिया होगा. सिर्फ मतलब ऐसे डिमोटिवेट होने से तो काम नहीं चलेगा. आपको वहां पे जाके उस दिन पेपर को फेस करना है, और उस दिन पेपर को फेस करने के लिए आपका अच्छी प्रिपरेशन होना चाहिए. अच्छी प्रिपरेशन के लिए आपको अच्छे से पढ़ना चाहिए. ऑल द बेस्ट.